Hello, my name is Marcus Sirigar, and this is my SSDE 8-week intensive summer research experience presentation. And over this last 8 weeks, I had the pleasure of being a molecular scatologist. Now, do you like strawberries and other fruits? Do you like seeing flowers at your garden or at your neighbor's garden or in the forest? Do you like to drink coffee in the morning to get you out of bed to be a responsible adult? Do you like to eat chocolate at the end of your day to reward you for being a responsible adult? Or better yet, do you like chocolate covered strawberries? If the answer to any of these questions is yes, then you can thank the hard work and efforts of pollinators. The pollinators come in many shapes and sizes, mainly the little ones from bees and wasps to moths and beetles to bats and flies and even butterflies. But the most unique one out of them all, to me, are hummingbirds. Now, I got to work with the Pollination Ecology and Conservation Lab with my lovely professor, Dr. Jenny Hazelhurst. And I got to work on one of her many projects, and specifically the Hummingbird Diet Project. Now, in this project, we wanted to understand the adaptation of hummingbirds with the change in their environment. And we did this by looking at Anis hummingbirds and Rufous hummingbirds. And we did this by looking in two main areas, in rural and in urban areas. In rural areas, they would have more natural, local flowers and sources of food. But in urban areas, you'd have, a lot less local, a lot less native plants that these hummingbirds would feed off of and pollinate. And we wanted to see how they differed. And we did this by collecting their fecal samples, and we were looking in the DNA for insect and floral remnants. And our goal was to meta barcode the genetic remnants in the feces to identify which kinds of flowers and or invertebrates were in these two hummingbirds. Now, my role is, as listed in the beginning, as a molecular scatologist, but I also got to work in the field, which was, I would say, 15% of my job, and then the other percentage was in the lab. When I was doing field work, I had the privilege of catching hummingbirds and collecting their fecal samples. I even got to hold hummingbirds, which I can say is pretty cool. But most of my job consisted of being in the lab where I processed these samples, where I would extract DNA from these samples and I would duplicate them using PCR. And I'd be looking for genes of plants and insects using RBCL primer and ZBJ primer. And then after all that, I would use gel electrophoresis to check for the presence of DNA. Now, my experience before the program was mainly just biology lab coursework like Bio 140 A and B, which had me get comfortable with using pipettes, uh, agros, and PCR. But I also had experience with organic chemistry 1 and 2, where I just got used to using chemicals of all shapes and sizes. Now, in the spring, I had the privilege of having an internship with the Pollination Lab, and I got to work with Dr. H and her lab mates. And I even got to work with a CSUEB grad student that was a molecular biologist. And it was really good to talk to him and to have an individual that I could almost use as guidance to what I was doing. And even though this was a 4-month unpaid internship, I gained a lot of experience that money almost can't buy. Within those 4 months, I just did basic PCR product and gel electrophoresis. And on this last image of the gel to the left is actually my first time getting successful bands in the lab over the spring. Super amazing. Now, my experience during this summer program kind of expanded what I was already doing, in that I would now do DNA isolation, where I would extract DNA from the samples, and then actually work with the master mix and make proper concentrations so that everything can work like cooking in a kitchen. I even made positive bug controls. So when I ran the gels using the ZBJ primer, that I could verify that the test was working. And when it came to gels, we would use something called TBE, and when we'd run out, we'd need more. So I got a chance to make those safely in a fume hood by scratch in the presence of Dr. H. And also with the gels, I got to adjust formulas, whether we're doing a 1% or 2% gel, and as well as the sizes of them. Lastly, on the latter end, I got to do data entry, which was new to me, but very much needed. Now, my experience at the end of the program just gave me a lot of confidence in what I was already doing. It gave me confidence in sample prep and DNA isolation and more PCR working with master mixes and positive bug controls and making now TBE, and lastly putting in data into the Excel sheet so we could see how many samples were successful, how many were unsuccessful, and where we can go from there. Now, my main challenges was no bands. And this, to go from the pieces of paper to this image took roughly about 30 hours. And if you work 30 hours and you have little to no results at the end, it can be a little daunting, but we keep our heads up and we trust in the process. But no bands would kind of come from either three main spots: DNA isolation, PCR, or the gel. And with DNA isolation and PCR, we could have been working with old chemicals. There could have been actually a lack of DNA in the sample, or easily the sample just degraded over time due to us taking them in and out of the freezer too much. When it came to PCR, could have made incorrect concentration to the aliquots, which would not yield positive results. And either we mixed it a little bit too much or not enough. And in doing so, wouldn't properly have the DNA attached to the primers and give me bands. And lastly, bubbles, when we were running the gel, would sometimes pop up, and this was due to either old TBE, which is why I made new ones, or from soap residue from somebody cleaning it with soap. And lastly, data cleanup from multiple people putting in information and sometimes getting confused. And here is a comedic sketch of PCR in a nutshell. Now, my accomplishments were many, but I would want to narrow them down to three main things, which was being able to make proper aliquots for master mix using mathematic skills that I gained from doing chemistry, successfully producing positive bug controls where I had to go out and catch bugs on campus, 9 different ones, bring them back, freeze them, remove their guts, DNA isolation, and then PCR and the gel, which I did successfully. And probably one of my major accomplishments was being able to use the large gel with about 36 samples. And I believe I also did one up to about 45 in comparison to our normal 15 samples. Now, benefits for future CSUEB students to take this summer program is that this is a real lab experience. This is not your 2 hours twice a week kind of experience. This is 20 hours a week of being in this lab. And you get to have a one-on-one experience with this professor in the lab versus 20 on one. And because this is not a rushed semester school year, you have time and space to fail. And I think that is probably one of the most important points. And on a more relaxed setting, that being around professionals in the field, not during all the commotion of school, really allows you to pick their brains properly. Now, some of the challenges are that this is a part-time position. With part-time, you're getting limited experience and limited income. And upon doing this whole program, you may find out that you may or may not like what you're doing or the research project or the professor. And this is where I would have tips for future CSUEB students going into the program. I would try to secure a position in the spring in the lab that you want to work for. Even if it's unpaid, you get to have this experience that money almost can't buy. And also, this will confirm your interest in the research and also that you can get along with this professor and vice versa. I would also say be involved with all aspects of the research project, both beginning, middle, and end if you can. Read peer review articles that are relevant to the research project because it'll put you in that scientific mindset, and then you will also be on the same page with your professor with the research. And always, always, always ask questions, whether you think they're dumb or smart or silly, ask away. This is the time. And be okay with failure. It is essential that you get this kind of in your brain and you're okay with it now because the more you fail now, the less you'll fail later. And luckily, this is not a rush setting, so take your time and enjoy it. Suggestions I would have is that income is limited. You are only working 20 hours a week, so do not expect to make a living off of this. You should probably find a secondary job, and you should probably plan for East Bay to pay you once a month. And actually, the first check comes in 6 weeks, whereas the second check will come in about 4 weeks after that. And if you can, talk to as many people as possible in your field. Talk to grad students, talk to undergrad students, talk to professors, talk to lab techs, because you never know who knows who to get you in the door for where you want to go. And lastly, make sure you're interested in the research, or at least in the process. And then I would like to thank Dr. J, Dr. H, Dr. V, and Matt, or Fernanda, Destiny, Naya, Sabina, Jakari, Captain Cisco, and myself. Thank you so much.